Thank the organizers for giving me the opportunity to talk to you about my research. We started working on Huanlangbing or HLB disease about 5 years ago for a couple of different reasons. The industry members approached our lab. We traditionally have worked on bacterial-plant interactions, and I was really intrigued because it's a very devastating disease, a very important disease. It's difficult to work on, and I like a challenge. And also, I think because the pathogen resides in the phloem and specialized cells within the phloem, I think that there's a lot of interesting biology we can learn by studying this disease. So in my group, we study really 3 different facets of Huanlangbing. We're interested in the HLB-associated pathogen, Candidatus Liberibacter asiaticus, or CLAS. We want to know what its genetic and phenotypic variability is. We're also interested in how it can actually manipulate its plant host. And then what we've been focusing on for the last couple of years is genetic improvement of citrus defense. So can we boost and improve citrus immune responses in order to decrease disease severity? So Carolyn mentioned the impact of the disease, but I just want to highlight this again. These aren't the latest numbers for Florida. It's a little bit over 80% that's lost as of 2019, but there's really been a significant decrease in Florida citrus production since 2005 because of the disease. In California also, there has been a linear increase in the amount of disease positives that have been detected using PCR validation for CLAS. And so the first disease tree was detected in Southern California in the city of Hacienda Heights in 2012. And there weren't any subsequent positives since then for a couple of years. And then in 2015, there started to be a pickup in the number of positives. These are all in backyard citrus, but because the insect vector is well established throughout the state of California, presumably the disease is already in commercial citrus groves, or it's very unlikely that we won't be able to completely stop the ingress of the disease in commercial citrus groves. And this is important not just from a health perspective, but also from an economic perspective. So California's citrus industry is very important economically for the state and the people of California. It's about a $3.4 billion industry with a total economic impact of over $7 billion. So it's been estimated that if even a 20% reduction in California citrus acreage, which could easily occur with a disease, would result in over 7,000 jobs that are lost, $127 million in employee income that's lost, and a $500 million reduction in state GDP. So it's a very important commodity for California. And so one question that we had was, where is this pathogen coming from, and what's the diversity of the pathogen in the United States? So we embarked upon a genome sequencing effort, and we sequenced a number of different bacterial from trees and psyllids collected in Florida, Texas, California, Mexico, and also Asia. And we also, in this data set, collated existing sequences for CLAS that were deposited in public databases. And looking at the phylogeny based on core orthologous genes, we can see that the population in the United States is not genetically uniform. If we look at Texas, this appears to be the most genetically uniform, and it clusters with bacteria that were sequenced from Mexico and also psyllid from Florida. If we look at California, it appears, at least from this analysis, that there were likely at least 3 different introduction events. And this is consistent with other research that's looked at prophage diversity in different bacterial samples. So pink is California. The original Hacienda Heights positive tree that was sequenced is shown here, and it clusters with bacteria from Asia. It does not cluster in the same region as a Hacienda Heights isolate that we sequenced in 2016, and it also does not cluster with isolates from the neighboring cities of San Gabriel or Anaheim. And so I think this is important to think about because not only are you probably going to have diversity in response to citrus variety or citrus genotype, but you're also probably going to have diversity of responses and pathogenicity from different bacteria. So one question that we have in my group is how does CELAS actually manipulate its plant host? And it can likely do this through a variety of different approaches. One mechanism that we're really interested in is secreted bacterial proteins called effectors. And we became very interested in these secreted effectors a couple of years ago because of some really excellent work out of Saskia Hogenhout's lab. She doesn't work on Liberibacter, but she works on phytoplasma, and I think it has some similarity to Liberibacter. So this is also a small bacteria. It's transmitted by a piercing sucking insect called a leafhopper, and it resides in the phloem, and in particularly in these sieve elements in the phloem of the plant. So these are the sugar-conducting elements of the plant. And the pathogen is limited to these sieve elements, but it can secrete proteins directly outside of its own cell. And these proteins are small, so they can move away from the site of infection through cell-cell junctions called plasmodesmata and influence disease severity. And what's really interesting, I think, about phytoplasmas, I think plant diseases are fascinating in general, but phytoplasmas will severely remodel plant architecture, and this occurs to some extent as well in Liberibacter-infected plants. So this is one example of phytoplasma infection in lime. It causes witch's broom in lime. You get a lot of auxiliary shoots. The reason for this is that since this is a vector-borne disease, this attracts the insect vector and facilitates transmission. And if you just make a Arabidopsis plant that expresses a single sect-dependent effector from phytoplasma, SAP11, you could remodel Arabidopsis architecture to phenocopy witch's broom. So we were interested in, what are the effectors from Liberibacter and how conserved are they? So Shri, a postdoctoral scientist in my lab, started looking at the different genome sequences that we have of Liberibacter. And what was actually surprising to us was that the majority of the sect-dependent effectors are conserved across diverse strains. And so this is actually fairly unique for a bacterial pathogen. Usually there's a lot of variation in effector repertoire, but not necessarily for Liberibacter. There's a few that are variable, but about 27 are absolutely conserved from one bacterial to another. So next what we wanted to do was to determine what the expression pattern of these effectors are in two different citrus hosts. And I'll talk about here, this is what we see in sweet orange, but we see some slightly different patterns in other genotypes. So we inoculated sweet orange by insect feeding and then took samples when we saw symptomatic plants. And we also profiled the expression of these sect-dependent effectors in the insect vector as well. And what we could see is that there are different suites of effectors that are primarily expressed in the plant host versus the insect vector. And we hypothesized that potentially CELAS is secreting effectors in order to differentially modify a particular organism that it's associated with. If we look at the size of these effectors, they're all pretty small. So most of them are less than 30 kilodaltons in size. And these plasmodesmata cell-cell junctions in plants should be able to accommodate many of these effectors and allow them to move from distal sites from where the bacteria is located. So we wanted to test this, and we wanted to test this using a effector called SDE1, because we know SDE1 is primarily expressed in the plant host. So we generated an antibody against SDE1 that can specifically detect this bacterial effector protein. And then we looked in citrus trees. And so citrus trees are large. If they're full grown, they're much larger than me. And they can live and be productive for decades. So we looked in trees that had regions that were symptomatic and regions that were asymptomatic. So what happens during disease is after transmission of the pathogen will sector in the plant. And so one region of the tree may be infected, but the entire tree will not become infected and succumb for sometimes a couple of years. So we looked in symptomatic regions. We can see clear SDE1 expression. We can detect the pathogen by qPCR. If we look in asymptomatic regions of the same tree, we can detect the presence of the effector, but we can't detect the presence of the pathogen. And so this indicates that potentially some of these effectors from Liberibacter can move away from the pathogen location. So what I've told you so far is that during insect feeding, the insect will deposit Liberibacter into the phloem while it's feeding during salivation. And then Liberibacter has the capability to secrete proteins outside of its own cell, and many of these are small, so they should be able to move through cell-cell junctions to adjacent sites and even farther than that within the plant. So another question that we had is, what are the functions and the host targets of some of these Zealots effector proteins? And this is a collaboration between a senior graduate student, Jessica Franco, in my lab, and also Professor Wenbo Ma at UC Riverside, and a previous graduate student in her lab, Kelly Clark. And we focused on SDE1. One of the reasons for that is we know it's conserved across diverse bacterial samples, and it's conserved across Candidatus Liberibacter asiaticus. And we can see that it's predominantly expressed in plant hosts. So citrus or periwinkle, you have high level expression of SDE1, but much lower level expression in the insect vector. So we performed a yeast-2 hybrid screen to identify potential interacting proteins for many different effectors. And what we found for SDE1 is it could specifically interact with a variety of different proteases. And we were really interested in this result because these proteases belong to a family called papain-like cysteine proteases, and these are known to be immune proteases in other plant systems. So just kind of highlight what they do. Of course, nothing's known about them in citrus, but we do know from some nice work in Arabidopsis, maize, and tomato that upon perception of a pathogen as non-self, these proteases are induced at the transcript and protein level, and they can either directly target pathogens or they can amplify defense signaling. And so not surprisingly, there have been other pathogens that have secreted either proteins or small molecules that can directly inhibit these immune-related proteases in plants. So first we wanted to figure out if these PLCPs are actually induced in response to infection. And so we have an antibody that was generated against a PLCP called AALP. It's an immune-related protease. And what we did is we took a branch that was either symptomatic and had high titer of bacteria, asymptomatic with low titer, or uninfected, and we blotted that on a nitrocellulose membrane and processed that like a Western blot. And so if you think about where the phloem actually is within the stem, it's in this circular region on the exterior. And that is roughly where we see the induction of these defense-related proteases that increases with the presence of the pathogen. So we also purified recombinant SDE1 protein and tested for the ability to inhibit PLCPs using a fluorescence assay. So decreased fluorescence indicates decreased protease activity. And we can see here that SDE1 can inhibit protease activity in a dose-dependent manner. So can E64, a known protease inhibitor. We also wanted to see what actually happens in citrus. And so in collaboration with Nian Wang at the University of Florida, he generated transgenic navel plants that are expressing SDE1 protein. We can see that these transgenics can express the effector protein. And then we performed activity-based protein profiling for the proteases. So here, any band will indicate active protease, and the strength of the band indicates the strength of activity for that immune protease. So we can see that wild-type untransformed controls have strong protease activity, but we have decreased protease activity in SDE1 transgenic lines. So that indicates that SDE1 can directly inhibit PLCP activity both in vitro and in vivo. Our other question was, what actually is happening in the field? And for this, we collaborated with Veronica Anconda. She is an extension specialist at Texas A&M University, and she's located in Kingsville, Texas. And what they have access to in Texas is there are a number of different citrus groves. The pathogen is there in Texas. But adjacent to that, these are mature citrus groves. They're at least 20, 30 years old. There are also adjacent screen houses. And so these screen houses have trees that are located inside, but they're constantly screened for the presence of the vector and pathogen, and so these trees are disease-free. So we took healthy trees and symptomatic trees from the same grove, performed protein extraction and mass spectrometry to look at protein abundance, as well as performed activity-based protein profiling to look at the activity of these immune proteases or protease activity. And this is all done in sweet orange. And so what we can see is on the x-axis are the different classes of papain-like cysteine proteases, and on the y-axis is the level of activity or the intensity by mass spec. So we can see that if you look at uninfected versus infected for this CP3 protease, you can see an increase in abundance, and you can see a concomitant increase in protease activity. The same thing for XCP1. There's a decrease in abundance and a decrease in activity during infection. We see the same thing for AALP, an increase in abundance and an increase in activity. But there were three members of the SAG12 papain-like cysteine proteases that we can quantify where they did increase in abundance in each case, but they didn't change in activity between infected and uninfected. And so we currently hypothesize that these might be the main targets of the SDE1 effector, and we're following up on this work. So our current model of what's happening is that after CLAS is deposited into these sieve elements in the phloem, the plant is able to sense the presence of the pathogen. It can secrete immune-related proteases that should be able to amplify defense responses as well as potentially directly target the pathogen. But the pathogen is also able to secrete effectors outside of its own cell. These effectors, in some cases, can be mobile. And one of these effectors, SDE1, can directly target papain-like cysteine proteases and inhibit their activity. So even though there is a response by the plant side, it's clearly not enough to completely clear infection. So there are no cultivated citrus varieties that are disease-resistant to the pathogen. So this kind of brings me to our next area of interest, which is genetic improvement of citrus defense. And the work that I'll talk about is primarily performed by Tanya Taruno, a postdoctoral scientist in the lab, and was initiated by Jessica Franco, a graduate student. And so here, we're trying really three different approaches. One is genome editing of pathogen targets or negative regulators of resistance. So we're targeting some of these PLCPs. We have other effector targets that we're targeting using genome editing as well. And this is a collaboration with Mian Wang at University of Florida. We're also really interested in what are these immune responses in diverse genotypes, so citrus and wild citrus relatives. What's the repertoire of immune perception in citrus? And can we utilize diversity in immune perception for genetic improvements, either as a tool to accelerate traditional breeding or for use in biotechnological approaches? So I'll talk about the last two here. So if we think about citrus, we know that it's actually pretty phenotypically as well as genetically diverse. So citrus is a member of the rue family, or Rutaceae, and there's 160 genera in the Rutaceae family and over 2,000 species. This can be then divided into subfamilies, so that we have the Orontoideae, the Rutoideae, and the Totaloideae subfamilies. Citrus is present within the Orontoideae subfamily, and there's 33 genera there and many different species. And then these subfamilies are then subsequently divided into lineages or tribes phylogenetically. And citrus that we eat is within Citrinae tribe, but there's also Clausinae and Balsamocitrinae. So all sexually compatible cultivated citrus species and their wild relatives would be present within the Citrinae tribe. And we also know from some research out of multiple groups, including our collaborator Chandrika Ramadugu, who published this paper in Plant Disease, that there's a spectrum of HLB development in citrus and wild relatives. So sweet orange is the most susceptible, like Valencia or Washington navel. The sweet orange will succumb to the disease. There's very high titers of the pathogen present. But then there's this gradient where you have some forms of citrus relatives that are not as susceptible, like Australian finger lime, which is like the caviar, I guess, of citrus that is used. And then there's also other regions where there's genotypes where there's really no CELAS that's present. And so that can either be because the vector does not feed, or in some cases, like Australian desert lime, the vector will feed, but the pathogen is cleared and disease will not develop. And so we were interested in these genotypes more on the extreme end of no CELAS, because that suggests that potentially there could be active clearing of the pathogen, and maybe we can use this information for genetic improvement. So if we think about plant immunity, plants have innate immune systems, so they actually can have thousands of germline-encoded receptors present in their genomes, but they don't have adaptive immunity like you and I do. So there's no specialized circulating immune cells. There's really considered to be two main receptor types that can perceive pathogens. There are these pattern recognition receptors that are displayed on the cell surface. These have some structural or domain architecture similarity to toll-like receptors, and they perceive either pathogen-associated molecular patterns that we also call microbe-associated molecular patterns, PAMPs or MAMPs. As non-self, this will trigger a cascade of defenses inside the cell. It'll also trigger the production of reactive oxygen species outside the cell that's directly antimicrobial and involved in cell-cell signaling. There are also immune receptors inside plant cells that are NLR receptors that can directly recognize pathogen effectors or effector activity and trigger robust defense. So we were most interested in these pattern recognition receptors that are displayed on the cell surface. And the reason why is because there's some precedent for MAMP perception from piercing sucking insects. So by now you've seen this image of the psyllid feeding at a 90-degree angle on citrus. But before I worked on the disease, it looks vicious up close, and it is a vicious insect. But it's also extremely small, okay? And this causes a lot of problems for control of the pathogen, because if you want to spray with an insecticide like a neonic, how are you going to actually be able to reach the psyllid, which is thigmotrophic and likes to squeeze between little spaces, okay? So here it is feeding, okay? And when the psyllid feeds, it will insert a stylet into the plant, and it's going to then taste different cells and salivate while it's doing that. And that saliva can contain psyllid features that can be perceived, as well as any bacterial MAMPs that could be potentially perceived, okay? And so it's going to taste here. This is where you can get defense that's elicited that could control the disease. And then once it reaches the phloem, that's when it will actually start to feed. So it reaches its preferred cell type, okay? So here you can see the psyllid feeding, and when it's in the phloem and stylet reaches the phloem, it wants to feed on the protein in the phloem, which is the minority of the phloem. So most of it is sugar, protein is like 2-3%. And so it'll secrete this waste called honeydew, which is a sugary waste product, and that will also elicit immunity in plants. So we think that if MAMP perception would actually work against CELAS, this would have to happen during natural infection, and it would have to happen very early on during infection. So first what we wanted to know was what's the response of citrus and citrus relatives. So we synthesized a variety of different MAMPs from bacteria, and also those from insects like chitin that could be perceived by other plants. Then we spent quite a bit of time trying to develop assays for MAMP perception and immune assays for citrus. So if you think about citrus, there's a lot of different leaf morphologies. So these are all cultivated, right? I just put the common names: navel, grapefruit, lemon, mandarin. These look okay, and they're not difficult to assay. But then you have some of these more exotic wild relatives, and they have very diverse leaf morphology, and it's more difficult to develop assays for those. So one of the assays that I'll talk about today is the assay for reactive oxygen species. So we optimize this for different genotypes, where you can basically take leaf punches, elicit with a MAMP, and then use a luminol-based assay for the presence of reactive oxygen species. So then you get a plus or minus for recognition. So we looked at about 90 different species of citrus and citrus relatives, and I won't show you all the data, but what we did find is that there's a wide diversity in MAMP perception across citrus genotypes. So here are the more resistant relatives. Here are the susceptible varieties and other susceptible relatives. And what we can see here, if we're just looking at chitin perception, that there's a variety of different responses just in these few species that I'm showing here. And not only is there a difference in presence and absence, but there's a difference in response magnitude as well. The other thing that we noticed when we started to look for bacterial MAMPs to test is that CELAS can actually mask multiple immunogenic MAMPs. So it has polymorphisms in its region of flagellin that's immunogenic on plants, and so that didn't give us a robust response in any of the citrus genotypes we tested. It also, another common bacterial protein that could be perceived by plants is cold shock protein, and there's a 22 amino acid immunogenic epitope there. And that also has multiple polymorphisms in CELAS, and that masks its perception in many genotypes. So you can see here's canonical bacterial CSP22. You can see perception in many of the more tolerant or resistant genotypes, and then here's CELAS CSP22, and it can only be perceived out of 90 different species we tested in 2. So we hypothesize that there's probably a novel immune receptor there that's responsible for it. So there were these 2 citrus genotypes that we found that actually respond to most tested MAMPs, and that's Uganda powder flask and Chevalier's eglypsis. These are members of the Balsamo citrinae tribe, and they are able to be fed on fairly well by the insect, but the pathogen does not cause a significant disease, and so it can recover from CELAS infection. So what I've told you so far is that sweet orange is unable to perceive tested CELAS and insect features. So if you were looking on the resistant to susceptible, most of the susceptible sweet orange was always on the right side, and so that didn't actually respond to most of the features that we've tested. So we want to use this information to try to move forward, and one way that we're thinking is to try to transfer stacks of receptors for bacterial and insect sensing into these elite citrus types for crop improvement, and then test those resulting lines for enhanced resistance to HLB as well as other diseases. So it's not just Huanglongbing that's a problem for citrus, Phytophthora, Xanthomonas citri. There's a number of other pathogens that are fungal, bacterial, oomycete pathogens that are problematic for this citrus production as well. And so towards testing these stacks, we're collaborating with Jim Thompson at USDA at the PGEC Center. And so what Jim has developed is he's developed a stacking system in Agrobacterium where you can stack multiple genes for transfer into citrus into a defined genetic location. And these can be transferred all at once. So not just taking one gene, but taking 5 or 6 genes in tandem and putting those in. And so we're doing that. We have a stack for insect sensing, a stack for bacterial sensing. We have a large stack combining bacterial and insect sensing, and then we've combined many different receptors that could potentially be important for bacterial sensing as well. So we've cloned all of these, we've put them into Agrobacterium, and now we're waiting for the transgenic plants. The other question that we had is whether we can actually use this as a breeding tool to rapidly identify promising genotypes. And so we're not breeders ourselves, but we're collaborating with breeders at UC Riverside and University of Florida. And so they're using this as a pre-screen to identify promising genotypes, because it's not hard to have thousands of citrus seedlings available every year. What's difficult is growing those up and testing them and figuring out what's promising and what's not promising. So we're using this kind of as a first test. And then finally, we're also looking to try to identify immune receptors using a homology-based approach. And so it's known from other plants that you have these pattern recognition receptors that can perceive each one of the features that I've tested. So we're looking for homologs to those particular receptors to see if they will actually function as a bona fide immune receptor in citrus. And so I'll tell you a little bit about that. So Tanya Taruno is leading this work. So many of these different citrus genotypes have been sequenced, and some of our collaborators are generating very high quality genome sequences for these citrus relatives. And so we're interested in the chitin receptor. And so the chitin receptor is known to be Lic5 in Arabidopsis. And we identified the closest homolog to Lic5 from citrus. And here you can see the phylogeny of the different citrus relatives for Lic5. And then what Tanya did is she tried to complement the Arabidopsis Lic4, Lic5 double mutant, so this is unable to perceive chitin, with the citrus receptor. And so this is preliminary data, so this is the first generation, but it seems promising, and I think the time will tell to see if we can do this for other receptors. But basically, any transgenic plant where we had robust expression of the receptor, we were able to see response to chitin in some of our immune assays. And so we're testing this now in subsequent generations. So to summarize, I've told you that different CELAS bacteria have genetic diversity within the United States and worldwide as well. Texas appears to be the most uniform, but really we need to get more and more sequences to truly say that it's a uniform population. Florida is the most diverse, and California, it looks like there are at least 3 separate genotypes that are available and are currently circulating, and there may be even more than that if we increase our numbers. And so I think that representative bacteria should be used to test promising plant material. So not only look at different plant genotypes, but also look at representative bacteria from a particular geographical region. I also talked about CELAS effectors and how they're differentially expressed in vector and host, and this might give us some clues as to what their function might be. We have evidence for effector movement through plasmodesmata, and we know that the SD1 effector can target citrus immune-related proteases likely to dampen defense. And then finally, we started a project looking at immune receptor diversity and perception in different citrus genotypes and citrus relatives. If we look at resistant genotypes, they can respond more robustly to tested MAMPs compared to cultivated relatives. In particular, sweet orange appears to be a real dud for immune perception. And we're hoping that immune receptor diversity can be harnessed for genetic improvement, and we're just starting to do some of those experiments now. So I'd like to thank the people that have done all of the work. This is my lab at UC Davis. I'm fortunate to work with a talented group of international scientists from diverse backgrounds, which I think really helps move the science forward. The people that worked on, on this project were Tanya Taruno, Jessica Franco, and Shri Thapa, and then also our lab tech, Tanya Chilcote. And then I'd also like to thank our collaborators. So we have a number of collaborators at UC Riverside. Most of the citrus in California is grown in Southern California. And there they have the citrus variety collection where a huge variety of different citrus genotypes and wild relatives. And so we collaborate there with Chandrika Ramadugu for breeding, Wenbo Ma for effector work, Georgios Vidalakis, James Borneman, and Neil McRoberts for outreach. We also collaborate with Nian Wang for genome editing, Veronica and Conda for access to field samples. Jim Thompson is doing the receptor stacking, and then Rainier Vanderhorn from Oxford is an expert on PLCPs and has helped us with those assays. And Kranthi Mandati at Texas A&M is trying to see if we can actually use these perceived microbial peptides as maybe a treatment option for either pre-disease or post-disease to try to clear the pathogen. And I didn't have time to talk about that today. And then also funding from USDA NIFA, the UC Multi-Campus Research Program, and previous funding from Citrus Research Board. Thanks.